Hello everyone, and welcome to today's live webinar, Protein Concentration Assays, Novel, Simplified, and Rapid Techniques for Quantifying Proteins in Solution, presented by Ramesh Ganapathy, a Research and Development Scientist 3, Protein and Cell Analysis at Thermo Fisher Scientific. I'm Christina Jewell, and I will be your moderator for today's educational webcast, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process of attaining your credits. Please join me in welcoming Dr. Ramesh Ganapathy. I will now turn the presentation over to him.
Hello, thank you for that introduction. I'm going to be talking about protein concentration assays, novel, simplified, and rapid techniques for quantifying proteins in solution. So this is going to be the outline of my talk. It's going to be principles of protein concentration assays, the purpose and selection of standards, types of protein assays, and factors influencing the selection, an overview of peptide assays, instrumentation, and technical resources and support. Let's begin with what is a protein assay. Simply put, it is a method to determine concentration of protein in a solution. Now, what we need in an ideal protein assay is that it should be fast, it should be reproducible, it should be accurate, sensitive, non-interfering, should have a broad range, and it should be safe to use. Traditional assays contain limitations with respect to these parameters, but recent innovative developments provide near ideal solutions for protein concentration determination. There are two ways to determine concentration of proteins. One is an old approach which is based on Beer's law. Now, due to the presence of aromatic amino acids such as tryptophan, tyrosine, phenylalanine, proteins demonstrate an absorbance at 280 nanometers. By measuring the absorbance at 280 nanometers, concentration of proteins can be determined provided we know the extinction coefficient of each protein. Therefore, this is an absolute method of protein concentration determination. The relative method of protein concentration determination is by using a standard curve. Here, you do not need to know the extinction coefficient of the protein. What you will need is to have a standard protein, plot a standard curve based on the known concentration of that standard protein, and measure the concentration of the unknown protein based off on that standard curve. Now, for the relative standard curve method, we have a copper-based method and dye binding method. I will get into more details of these methods in the following slides. Now, for Beer's law, the aromatic side chains absorb light. Their absorbance can be measured at 280 nanometers. Now, the equation is absorbance is extinction coefficient times the path length times the concentration. So from this equation, we can see that the absorbance at 280 nanometers is directly proportional to the concentration. If your extinction coefficient, that is epsilon, is known, the absorbance 280 nanometers is known, and the path length, which is usually 1, but which can change from instrument to instrument, is known, then you can calculate the concentration by plugging all those values into the above equation. Now, for the standard curve method, we have the copper reduction method, in which copper 2 is reduced to copper 1 by protein. The copper 1 is then chelated by a chelator and color is produced. In this example that we have shown here, violet color is produced, which has an absorbance at 562 nanometers. The second method is the dye-based reaction, in which proteins bind to dye in acidic conditions, due to which the dye solution changes color. In this example shown here, a blue color is formed, which has an absorbance at 595 nanometers. Standard curve. So how do we select the right standard? First, the standard must be of known concentration. The standard must be close to the sample, as close to the sample as possible. Now, for protein samples, a very prevalent standard is BSA protein. It is available in 2 mg/ml ampules from Thermo Fisher Scientific. And for a single purified protein, if you want to determine the concentration of a single purified protein, for example, scientists and researchers use that in binding studies in chromatography, then it is advisable that you use that same protein as the standard instead of using BSA. However, if you have complex samples, protein mixtures, lysates, it is a good idea to use the BSA as a standard. For antibody or IgG samples, BGG is a popular standard, and that is also available in 2 mg/ml ampules. Now, for peptides, we have a proprietary peptide digest, and this is unique to Thermo Scientific, which can be used as a standard for determining the concentration of peptides in solution. This is available in 1 mg/ml concentration, and I will talk about peptide assays later in the presentation. How do you use a standard curve to determine the unknown concentration of protein complex sample? Here we have a graph that has BSA concentration on x-axis and absorbance in its y-axis. We generate a standard curve, find out the absorbance of the unknown sample, which is typically measured at the same time that the standards are measured, so that there is no experiment-to-experiment error. Now use the equation of the line to determine the concentration of unknown from its absorbance value. You have to keep in mind that if the sample is diluted, then your final concentration of the sample is determined by the concentration, which is obtained from the graph, times the dilution factor. Now, this is a typical workflow for a protein concentration assay, where you prepare and pipette samples and standards. You prepare the working reagent mixture. For example, for the BCA assay, we mix copper solution and reagent, and I will explain in detail about the working reagent for BCA in the next slides. You add your working reagent to each sample and standard, incubate for the allotted time, and read the absorbance at specified wavelength. Once the protein assay is done, we can then perform downstream applications such as Western blotting, mass spectrometry, and protein purification. I'm going to go in this slide from left to right to compare each assay across each parameters. The parameters that I'm going to talk about here are reaction mechanism, the type of assay, the time, temperature of incubation for each assay, the standard curve, and then the compatibility. Now for the BCA, amino acids such as cysteine, cysteine, tryptophan reduces copper 2 to copper 1. More importantly, peptide backbone also reduces copper 2 to copper 1. This in turn chelates with BCA chelator and changes the color from green to violet. As the protein concentration increases, your violet color intensity also increases. For the dye-based assays, such as the 660 nanometer assay and the Bradford assay, positive amino acids such as arginine, histidine, and lysine, which are facilitated by the low pH of the protein assay reagent, deprotonates the dye. Upon deprotonation, the color of the dye reagent changes. In 660 nanometer assay, it goes from reddish orange to bright green. In Bradford, it goes from reddish brown to blue. Now, in the BCA, the working reagent is made up by mixing copper sulfate solution to the chelator and is then added to the sample. In dye-based assays such as 660 nanometer assay and Bradford assay, the reagent is a simple mix and read reagent, so it is directly added to the sample. The time and the temperature of incubation is also different for each of these assays. In BCA, the incubation time is 30 minutes at 37 degrees C, which has been reduced to 5 minutes with no heating with our new product called as a Rapid Gold BCA. I will talk in detail more about this new product in the next few slides. Now, BCA and 660 nanometer assay have a wide dynamic range, which goes from 20 microgram per mil of sample to 2000 microgram per mil of sample. The Bradford assay, on the other hand, has a narrower dynamic range from 100 microgram per mil to 1500 microgram per mil. About compatibility. BCA is compatible with detergents in your samples, but it is not compatible with reducing agents. This is because the reducing agent will reduce the copper from copper 2 to copper 1. So you will not know why the color change is happening. Is it happening because of your reducing agent, or is it happening because of your protein? However, to offset this problem, we have a product called as the reducing agent compatible BCA assay, where you can use reducing agents in your sample up to a certain compatible concentration. Our 660 nanometer assay is the most compatible assay than any other assays, and it is compatible with both reducing agents and detergents. The Bradford assay is not compatible with detergent, but it is compatible with reducing agent. However, we have a detergent-compatible Bradford assay, which is also compatible with detergents. I will share some data in the future slide to show how the detergent-compatible Bradford assay works. History of BCA protein assay. Now let me talk to you about our most popular assay, which is the BCA assay. We order a complete family of BCA assays, which include the traditional BCA assay, which is our most popular BCA assay, the micro BCA assay, which is for samples with very low concentrations, reducing agent compatible BCA protein assay, which is a BCA assay for samples with reducing agents in it, and now we have a new BCA product called as a rapid gold BCA, which gives the same sample accuracy as the BCA in 1/6 the time with no heating. Instead of waiting for 30 minutes at 37 degrees C, similar results to BCA can be obtained with this new product in 5 minutes at room temperature. Like I mentioned, BCA is our most popular assay. However, results take up to 30 minutes to obtain, and in addition to that, the reaction needs to be heated at 37 degrees centigrade. With this new product, the same results can be obtained at room temperature in just 5 minutes. Now the graph below shows the exact same standard curve obtained between the old traditional BCA, which is done at 37 degrees for 30 minutes, and with our new rapid gold BCA, which is done at room temperature for 5 minutes. So with this new product, we break traditional barriers. We get the same accuracy, the same low protein to protein variability, and I will explain what is protein to protein variability in the later slides. The results can be obtained with this new assay in just 5 minutes. So, upgrade your experience with the new Rapid Gold BCA. The gold bullets in this slide shows the difference between the traditional BCA and the Rapid Gold BCA. We kept the working reagent ratio the same, and we could get the same low protein to protein variability. However, as mentioned before, the time is reduced from 30 minutes to 5 minutes, and the temperature is reduced from 37 degrees C to room temperature. The color is different. Instead of green to purple, which we are used to seeing in traditional BCA, we have green to gold color with this new assay. As a result, the absorbance maximum, where you read the plate, changes from 562 nanometers for the BCA to 480 nanometers with this new assay. Now let me talk to you about detergent-compatible Bradford assay. As I mentioned, Bradford assays are not compatible with detergents. But 3 years ago, we came up with a Bradford assay that can be used for samples with detergents. Now in the graph on your left, you see that the blue line shows a curve of different concentration of BSA with no detergent. It works just like we expect it to. But as you can see, when we add detergent to the sample, the curves go all lopsided. But with our detergent-compatible protein assay, we can see on the right that all the curves merge and look exactly like the curve with no detergent. So for all these detergents at these different detergent concentrations in BSA, we have a good calibration curve with BSA standards. We at Thermo sell a lot of Lysis buffers, which are very popular, and using all these buffers, we see that our detergent-compatible protein assay is compatible with all of these Lysis buffers. So now let's discuss what are the factors that influence the selection of protein assay. One is your assay time, that is how long it takes to prepare the working reagent, and what is the temperature required for the assay. In addition, we also need to consider how long the assay takes to get the results. Then, what is in your sample? Do you have detergents, reducing agents? How much sample do you have? Is your sample a single protein, a mixture of protein, or a cell lysate? Is your sample a protein, a peptide, or an immunoglobulin? Then we have to consider linearity of the curve. Which assay gives you the accuracy at the high end? What is the dynamic range for each assay? What is the lowest concentration that you can detect with each assay? Also, we have to see which assay gives the lowest protein-to-protein variability and how much accuracy we get from sample to sample. So now let me talk a little bit about linearity and accuracy. As you can see in the BCA standard curve and in the 660 nanometer standard curve, we have a wide range which is from 20 microgram per mil to 2000 microgram per mil and an excellent linearity. However, when you look at the Bradford standard curve, we see that it has got a much narrower dynamic curve. Now, if your absorbance is anywhere between 1500 microgram per mil and 2000 microgram per mil, you are not going to know what the absolute exact concentration of your sample is because the curve is already saturated. And therefore, the protein assay reagent needs to be more linear, and therefore we have the Comasi Plus standard curve, where we have a new Comasi Plus reagent which gives you a much more linear curve and allows for better accuracy. Now let's discuss what protein-to-protein variation means. Depending on the type of amino acid present, their interaction with the protein assay reagent, different proteins at same concentration can give different responses. Ideally, we should see no difference in the responses. However, this difference, if we observe, it is called protein-to-protein variation, and it is undesirable. Now, copper-based assay gives the lowest protein-to-protein variability, whereas the dye-based assay gives the high protein-to-protein variability. The table below shows protein-to-protein variability measured with 14 different proteins. All of these proteins were made at 1 mg per mil concentration and their absorbances were measured. The ratio of the absorbance was calculated to the absorbance of BSA, which is why you see a value of 1 here. The percent CV of the ratios give protein-to-protein variability with each of these reagents, and here you see that the BCA type assays or the copper-based assay show low protein-to-protein variability and low CV around 15%. The dye-based assays show higher protein-to-protein variability. Now, considering all the parameters discussed, how can we choose the best protein assays? So, the dye-based assays give lowest time to results, that's good, than copper-based assays. Exception is the rapid gold BCA, which give results quickly. In general, copper-based assays are good for samples with detergents. in general, the dye-based assays are good for samples with reducing agents. The best linearity and low protein-to-protein variability in general is obtained for copper-based assays compared to the dye-based assays. Working range comparison. Protein concentration working range for various assays is shown in this figure. Here we see that the BCA, the rapid BCA, and the 660 nanometer assay as the best broad working range, which can go from 20 microgram per mil to 2000 microgram per mil. Bradford assays typically have a narrow working range, but I want to point your attention to micro BCA. Even though we have a narrow working range, with micro BCA, it gives us the best low limit of detection. We can measure as low as 0.5 microgram per mil BSA using micro BCA. So in summary, as you can see, we cannot get all the requirements from one assay. But what we can get is the best assay that satisfies most of the requirements. Our new Rapid Gold BCA, highlighted in gold, comes closest to being the most perfect protein assay. And to our knowledge, other than the fact that it cannot be used with reducing agents. It meets all the requirements that is required from a good protein assay. Now let us switch gears and move towards some specialty assays called peptide assays. For peptide assays, just like protein assays, we have two modes of concentration determination. Like measuring absorbance 280 for proteins that I talked about before, we can measure absorbance at 215 nanometers to determine peptide concentration. Colorimetric method is also possible to determine peptide assay, and that involves using micro BCA protein assay. However, this micro BCA kit is not designed to detect peptides accurately, and therefore here at Thermo Fisher we developed two powerful kits to determine the concentration of peptides. These kits were developed about 2 years ago, and I would like to share some data that we have generated using these two kits. First, let me talk about these two kits. One is a peptide quantitation kit, which is a colorimetric kit and uses the same principle as BCA, but it measures peptides. The other is a fluorometric assay, which uses a proprietary compound that reacts with N-terminus. Upon that reaction, a fluorescent molecule is formed which excites at 390 nanometers and emits at 475 nanometers. As concentration of peptide increases, the color intensity increases for colorimetric assay and fluorescent intensity increases for the fluorometric assay. So as you can see from this table, the fluorescent peptide assay has a broader range than the colorimetric assay, from 5 microgram per mil to 1000 microgram per mil for the fluorometric assay versus 15 microgram per mil to 1000 microgram per mil for the colorimetric assay. The sensitivity of the fluorometric assay is also better than the colorimetric assay. The sample volume is 20 microliters for the colorimetric assay versus 10 microliters for the fluorometric assay. The assay time is 30 minutes for the colorimetric assay versus 5 minutes for the fluorometric assay. One key feature of the colorimetric assay is that it can be used for TMT labeled peptides. We cannot use the fluorometric assay for TMT labeled peptides because it is based on the reaction for N-terminus. A key feature for the fluorometric assay is that it can be used to measure the concentration of single peptides. We do not recommend the colorimetric assay to be used for single peptides because the variation is a bit too much. As we can see for graphs from both fluorometric and colorimetric peptide assay, we get a very good linear curve up to 1000 microgram per mil of peptide. For the fluorometric assay, we see a nice linear curve when we use a digest of BSA protein or with a 1400 Dalton peptide called bacitracin. Now, bacitracin is also used as an antibiotic. The colorimetric peptide assay also gives us a very nice linear curve when used with increasing concentration of protein digest of protein AG or apotransferrin. As I mentioned earlier, we have developed a proprietary protein digest which can be used as a standard for peptides. Now, using that same standard to measure the concentration of 18 different digests, proteins and cell lysate digest, we used both the fluorometric and the colorimetric peptide assay and saw very similar concentration readings of the unknown sample. The blue bar is the concentration of the digest obtained using fluorometric peptide assay, and the red bar is the concentration of peptides using the colorimetric peptide assay. So depending on what type of assay you choose, you can be assured of very similar results with both of our peptide assays. Now let me talk about instrumentation for peptide assays. We offer two types of instruments: Nanodrop for small volumes as low as 2 microliters, and we also offer our regular spectrophotometers to read 96 well or 384 well plates. Now, how is the Nanodrop instrument used? This is a brief description of how this instrument can be used for a 280 nanometer absorbance measurement. You pipe it 1 to 2 microliter of sample onto the surface, you close the arm, the sample is drawn into a liquid column, optical fibers then transmit the light from a xenon flash lamp through the sample column. Absorbance at 280 nanometer is then measured. The sample is wiped away using Kim wipes, and the nanodrops have excellent dynamic range, due to which you don't even have to perform dilutions. Nanodrop spectrophotometer optics for protein assay measurements. So for the nanodrop 1-on-1, we need 1 to 2 microliter of sample. We can obtain a full scan from 190 to 850 nanometers. It has a touchscreen interface. We can use it for DNA, RNA, and proteins. It identifies contaminants and it gives corrected concentrations. As I mentioned, it's got a very high dynamic range and no dilutions are necessary. It's also got pre-programmed protein assay methods. I'll talk about that in the next slide. For higher throughput, we have the Nanodrop 8000. The amount of sample required is the same, which is 1 to 2 microliters. We can scan from 220 to 750 nanometers, and we can have up to 8 samples evaluated at the same time. The only thing is, with the Nanodrop 8000, we need to have a PC to operate. For cell lysate concentration determination, unlike a single protein, absorbance at 280 nanometers cannot be used. We can use any of the protein assays described earlier with the Nanodrop 1. This instrument has program methods for each assay. So you prepare your standards in your samples, you perform the assay like you would in a microplate or in a test tube, however you prefer. You click on one of the buttons which says protein BCA or protein Bradford, protein Lowry, or protein Pierce 660 nanometer, whichever assay that you have chosen to get the results. The program will guide you step by step for the addition of standards and the samples and for their measurements. The program then uses the equation of the standard curve and spits out the concentration of the unknowns without you having to do anything. Now this is our Varioscan Lux microplate reader. It has a built-in intelligence by which it can determine the optimal setting for each experiment. The Scanit software ensures that the instrument is simple to operate. It's very flexible and it can be used to measure absorbance, fluorescence, luminescence, and everything in one platform. So, as we have seen, there is no one protein assay that satisfies all our needs. Therefore, we have developed an interactive protein selection guide for customers where you can click on your sample type, click on your assay format, click on your sample composition. For example, if you have a detergent in your sample, you can click detergent. If you have reducing agent, you can click reducing agent. And what the program will do is it will choose the best protein assay for you. We also have a protein assay technical handbook that includes the advantages of each of the assay, the applications for the assay, the typical standard curve that you can expect with each of the assays. It has a detailed protocol. It gives you the mechanism of the reaction of each of the protein assay. It gives the detection range, the sensitivity, how low you can go in your protein concentration with each of the assays, and most importantly, it gives you a list of the interfering substances with its maximum compatible concentrations, which you can use for your protein assays. With this, I would like to take any questions that you may have. Thank you.
Thank you, Dr. Ganapathy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on the screen and click the send button. We'll answer as many questions that we have time for. Okay, let's get started. We have some great questions coming in today. Dr. Ganapathy, do you have a micro version of the Rapid Gold BCA protein assay?
No, not yet. But we are working on a micro version, and we hope that we could have that new product pretty soon, within a year. But right now, we don't have a micro version.
Okay, next question. My sample contains both detergent and reducing agent. Both the Detergent-Compatible Bradford and 660 assay should work, but is there one that's a better choice than the other?
They're both good. But like it was said in the presentation, we have pluses and minuses in both of them. The detergent-compatible Bradford has got a lower protein-to-protein variability, whereas the 660 assay has got a slightly better linearity. So depending on what you need from your assay, you should be able, you can choose one or the other. But yes, both of them are compatible with detergents and with reducing agents.
Thank you. Next question. Will the Rapid Gold BCA replace the standard BCA? For example, will the BCA be discontinued sometime in the future?
No, we do not plan to discontinue the BCA anytime in the future. We will have both these products.
Very good. Here's another one. More of a couple questions and a statement here. Are we only going to focus on proteins in solution? How about proteins adhered on surfaces? I would be interested in learning how to quantify proteins on surface.
Oh, this is a very good question. We do have a tech tip on how to quantify proteins on surfaces, specifically for magnetic beads, and there is no reason why you should not be able to do that on any other surface. Yes, we do have a tech tip, and you can use specifically for this particular example for the magnetic beads, we have used the micro BCA, and if you are interested, we can put, I can either put more tech tips on how to use the other assays, but there is no reason why you should not be able to do that using any of our protein assays.
Dr. Ganapathy, can you elaborate on why you're using dilution?
We are using dilution because, well, all of our assays only go up to 2 mg per mil. And so if your sample is more concentrated than 2 mg per mil, you will be falling off the linear portion of the curve. And so in order to get the right concentration of the protein, so that you should be able to measure it accurately and your concentration should be within the linear portion of the curve. When you're using a standard curve, you should dilute your samples. So what I would ask a customer to do is to dilute it 1 to 10, 1 to 25, 1 to 50, just so that you have a range, and then you can multiply that dilution factor to the concentration that you get from the standard curve.
Are any of these protein concentration assays amenable for using in the qubit?
Not right now, but watch this space soon. We are going to have a protein concentration assay which is going to be used with the qubit. Again, a very good question.
Dr. Ganapathy, we have an audience member who'd like to reference slide 18, and they're asking, regarding the Rapid Gold BCA, is it not compatible with reducing reagents?
Yes, that's true. It is not compatible with reducing agents because it follows the same principle as the BCA. Like I said, the reducing agent reagents will reduce the copper, so you will not know where the signal is coming from, whether it's coming from your reducing agent or whether it's coming from your protein sample. So it's not. But if you want to use a BCA type assay which is compatible with reducing agent, we have the reducing agent compatible BCA.
Now we're wondering, what is the maximum size of the peptides to use with peptide-friendly assays?
The maximum you can go up to 5,000 Dalton, even slightly higher. But I would suggest that for the colorimetric peptide assay, you can go a little bit higher in the molecular weight. But for the fluorescent peptide assay, I would stay within 3 to 4,000 Daltons.
Okay. Here's another question. Now my sample contains chlorophyll. Could that interfere with the quantification of proteins?
It should not. It should be okay. to use, again, it depends on what other things are present in your sample, and accordingly you should choose your protein assay.
Okay, we have time for about 2 more questions. How about, let's see, what's coming in. Do you recommend the Rapid Gold BCA for protein lysates and the RIPA buffer?
Yes, it's absolutely compatible with RIPA buffer. You can go ahead and use it.
Excellent. Okay, and we'll make this our last question. How about using these assays in simulated body fluid? What should I take into consideration?
There is no issue in with using it in simulated body fluid. The only thing you have to take into consideration is what other things are there in your sample. Like what I mentioned before, whether you have reducing agent, whether you have detergents, and accordingly you can go ahead and use any of these assays. Depending on their compatibilities, you could go ahead and use it.
Very good. I would like to once again thank Dr. Ramesh Ganapathy for his presentation today. Dr. Ganapathy, do you have any final comments for our audience?
Just that, we have a lot of protein assays. I know it's a big, broad choice, but I would just say that depending on what samples you need, what samples you have, and what is present in your sample, you can choose from a broad variety of protein assays that we offer. The Rapid Gold BCA in particular is a new product, and we have introduced something where you can get the same BCA type results within 5 minutes at room temperature, and that's a new product which came out last month. Just wanted to make a mention of that. Thank you.
Thank you, Dr. Ganapathy. And I would also like to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Now, before we go, I just want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again soon. Goodbye.
Bye.